Yeah, well, thank you so much again for the introduction and for organizing this symposium. It's been really great to hear a lot of talks. I'm learning a lot today about 2D materials and other things. But today I'm going to talk about mapping dynamic processes in energy materials with a technique called photo-induced force microscopy. But before I do that, I just want to acknowledge the people who did most of this work. So I lead a new group at the University of Utah. We're relatively small right now. We have about 3 grad students, 2 undergrads, and 1 high school student. And our group really focuses on understanding dynamic processes in unconventional semiconductors. So essentially semiconductors that aren't silicon. And so one thing we're really interested in is using electron microscopy to watch structural transformations in things like metal halide perovskites, which are exciting materials for solar cells. We also use a lot of scanning probe or AFM to watch dynamic processes in soft materials like polymers. And I'm going to focus most of my talk today on this work here with a little bit of perovskite stuff at the end, just because I was excited about 2D materials. And so the kind of big instrument that we have that does a lot of our measurements is the photo-induced force microscope that's made by a company, Molecular Vista. So this is an AFM that has the exciting capability that you can take nanoscale infrared images. So I'm not going to go into really how it works today, but you can really think of it as kind of shrinking down a Fourier transform infrared spectrometer to about 5 to 10 nanometer spatial resolution. So kind of taking that big bulky instrument that you used in maybe organic chemistry to look at molecular vibrations to basically map vibrations with nanoscale resolution. And so special thanks to the people at Molecular Vista. We're always adding new things to our microscope, and they've been very patient with us as we do this. And this is a picture of the microscope from our lab. And so our lab is really excited about dynamic semiconductor materials. So we think of semiconductors often as being static materials like silicon that we find in our computers. But I think the future of semiconductors is really trending towards the need for reconfigurable dynamic materials. And one area that I think is really exciting is in bioelectronics, where we need to kind of match the dynamic properties of biomolecules with electronics applications that can interface without perturbing these systems too much. Another area is clean energy materials. So this is an example of a solar cell that's on the roof of my building next to the football stadium at the University of Utah. And here we envision solar cells that maybe work really hard during the day, but at night can heal themselves through things like ion migration. And so the 2 kind of areas that my group is really excited about, one are organic mixed ionic electronic conductors. So these are materials that are organic. They're typically conjugated polymers, and they can transport both ions and electrons. And they're used for a number of kind of new devices like this organic electrochemical transistor, which I'll talk a little bit about later. And we're also excited about phase transitions in 2D hybrid materials, which I'll mention a bit at the end. And so these are materials that have inorganic and organic layers. And unlike the phase transitions that we saw earlier in the symposium, these undergo phase transitions by the organic layer essentially melting and becoming more liquid-like, which has important photophysical changes, which causes important photophysical changes in the inorganic layer. And we can capture this at the nanoscale and look at how we can tune the properties of these materials, essentially based on the organic layer. And for both of these systems, what we're really interested in is asking what are the molecular design principles. And so our philosophy is really, once we understand how these dynamic systems behave, then we can start to think about what kind of systems will work best for which applications. And so if we want to do bioelectronics, which materials can interface with biology most easily. And if we're interested in phase transition materials, we're looking at materials that can store things like heat. Okay, so just so that we're all on the same page, this is an example of an organic mixed ionic electronic conductor. So as I said, these are typically conjugated polymers. So they have this conjugated backbone that enables electron transport, and they have side chains that enable ion transport. So like these ethylene glycol-based side chains that you see here. And so these materials are exciting because they can transport both ions and electrons, but also there's a coupling between ion transport and electron transport in these materials, which makes them really useful for applications where you want to convert ionic signal to electronic signal and vice versa. And so some applications for these that I'm excited about are organic electrochemical transistors for bioelectronics, all polymer aqueous charge storage, so being able to make batteries or electrochemical capacitors out of these materials, neuromorphic computing, so having kind of brain-like computing architectures, and also thermoelectrics, where these materials are approaching the efficiency of inorganic materials and are much easier to fabricate. So a device that I'm really excited about is the organic electrochemical transistor. So this is the basic device shown here. It's very similar to a field effect transistor, only the ions can actually penetrate into the active layer. So instead of just staying at the surface and making a field effect, these materials have volumetric doping. So meaning that the ions actually go into the active layer and dope it in bulk. And so this gives us really high transconductance, but it also comes with a lot of challenges. And so the kind of cartoon picture of how these devices work is that you have your device in an aqueous electrolyte. So we have our ions in our electrolyte, and we put this essentially in an electrochemical cell. As we apply a bias, we inject holes or electrons from our underlying electrode, and ions then need to penetrate into the polymer layer to match with these electrons or holes to form these ion-polaron pairs. And so these ion-polaron pairs, they make the film more conductive, but since these ions take up volume, it also perturbs the local structure of our polymer. And so we have really complex rearrangements of the polymer at the nano and molecular scale. And the important thing is that these materials are reversible. So if you apply a reverse bias or just turn off the bias, the ions will come out. And ideally, you'll be back to your original non-conductive material. And so if we look inside our electrochemical cell and actually watch one of these things operate, so here we can see that our polymer is electrochromic. So as we drive ions into the film, we change its electronic properties, and it goes from this kind of pink color to this gray color. And so as a postdoc, I made a lot of these materials. We were able to test them on some kind of model systems. This is an example of a Venus flytrap with our device on it. And you can see as we poke at these trigger hairs inside of the flytrap, we can trigger these potentials that we can then record on our device. But what I'm going to talk about today is a little bit more of kind of a fundamental study that we did on these materials, looking at this process of how this ion essentially migrates into the polymer matrix, what happens to the polymer structure when it does this, and then how does this ion-polaron pair that we form actually move through this polymer matrix once it's formed. And for this experiment, we're comparing two different polymers, this P3MEEMT, which has a thiophene backbone and this glycolated side chain, and this PB2TTEG, which has a thiophene and phenylene backbone with these ethylene glycol side chains. So two polymers that look fairly similar. And the kind of spoiler here, what we found out is that when ions move through this first polymer, we have very minimal structural changes that occur. Yet with this PB2TTG, we notice that it converts between two discrete crystal structures when we inject ions. Okay, so at the beginning, we started with what we thought at least would be a pretty simple experiment. So we basically took a sample of our polymer. So this is a polymer thin film, and we electrochemically doped one side. So we essentially put ions on one side of our film, and we left the other side undoped, or having no ions on the right. And so what we thought would happen is that this interface would essentially migrate in time, and it would migrate according to Fick's law, where we would essentially see in the simulation a blurring out of this interface as ions move from the left to the right, and the absence of ions moves from the right to the left. And so we started with very low resolution optical microscopy. So here, just looking in an optical microscope at this interface over time. And what we essentially see is with P3M EEMT, a blurring out of this interface. So this is essentially what we expected. We think there's some fraction of immobile ions, which is why we see this kind of dark line that remains constant. But with PB2T-TEG, what we saw was something that really surprised us. So instead of a blurring out of this interface, we see that this interface migrates, but it stays sharp. And so we can simulate these results just using a kind of restricted diffusion model where we look at fix the diffusion constant to a certain concentration. But we wanted to really understand what was going on at this moving interface. And so here we wanted to go to much higher spatial resolution. So we wanted to take advantage of our photoinduced force microscope that will map out vibrational modes at the nanoscale. And so if we look at a nanoscale infrared spectrum, here we're using a infrared active ion. So this is with PF6. And so what we essentially do is we tune our IR laser to this PF6 vibrational mode, and then image so we can actually watch these ions move in our sample. And so I'm going to play a movie down here, which is going to show topography on the left and ion concentration on the right. And you can see as a function of time, we see this ion kind of wave flow across our thin film. And so you can see how these ions move across in our infrared image, and we don't really see too much here in our topography image. We can zoom in much further. So this is about a 10 by 10 micron scan. If we go down to 3 by 3 micron, we can now see this ion wave move across our sample. Here I'm adding this white line. This is just a 50% intensity threshold, which lets us nicely visualize how these ions move. And if you look really closely in the topography image, what you'll see is that it becomes a bit more wrinkled as this ion wave flows through our material. It's hard to see here, but if we switch now to looking at the change in topography, so now basically taking whatever frame we're on and subtracting the first frame, we can see this kind of wrinkling effect as the ions move through our material. And so the question is, okay, well, we can observe this change in the structure. It's obviously correlated to ion migration, but how is this structure actually changing? So we zoomed out again, and this time we're using grazing incidence wide angle X-ray scattering. So essentially to get a diffraction pattern of our polymer. And so we do this as a function of voltage. And so this is essentially as a function of driving more and more ions into our sample. And so at low voltage, we don't really have ions in our sample. We get this diffraction pattern here. And as we add more and more ions, we suddenly see a jump in the spacing between essentially our polymer chains. And after this jump, we get a completely different diffraction pattern. And so this was really cool to see. And I've shown these to people, and they've been questioning, is this just two different polymers? But what it actually is two different crystalline phases of the same polymer. So one without ions and one with ions. Okay, and so the next question is, okay, well, how are we getting from one crystal structure to another? What you could imagine is that this polymer kind of acts like what happens in a standard conjugated polymer, where as you add more and more ions, the polymer kind of slowly moves apart continuously. The alternative is that there's something that's more analogous to a structural phase transition, where you actually see a discrete change in the structure of these polymers. And this usually happens in inorganic systems, like those found in lithium-ion batteries, where as you inject lithium, you see this kind of discrete structural change. So we looked inside this gap here, and what we essentially found was a phase coexistence. So we saw two discrete phases based on these two crystals. So we know that both phases are present in this material inside of this gap. And so this means that we're undergoing a more discrete change between these two crystalline phases. And so this is very similar to what you see in lithium-ion batteries when you're charging them. And so in analyzing these spectra a bit further, we developed a model for how we think this might be happening. So we think the polymer is behaving almost like a molecular zipper. And so we start off with these interdigitated side chains. And when the ions enter the polymer essentially unzips. When it unzips, this actually causes the backbone of the polymer to crystallize, which gives us a completely different crystal structure. And this process is reversible. And so we can essentially zip and unzip the polymer just by driving ions in and out electrochemically. So we've been able to observe this phase separation in bulk, but now we want to understand, what does this actually look like at the nanoscale? And so again, we can turn to our photo-induced force microscope and actually image the polymer as a function of voltage. And so when we do that, this is what we get for the topography. You can see that very specific regions are swelling as we increase the voltage. And then we can also look in the infrared at our ion and see where the ions are in our film. And so when we do that, we get images that look like this. And you can see from these images here that the areas that are swelling of the polymer are essentially the areas where we're seeing ions. And so this was really exciting. We still don't quite understand this exactly why we have ions going to certain places and not other places, but it's still a work in progress. And so you can see here, if we look at change in topography, you can see these correlations may be a little bit better. And just putting this all together, you can see here as we go from our kind of undoped state with no ions to just changing the voltage slightly, we can see these kind of features form in our material. So this looks almost like a kind of nucleation and growth mechanism, where inserting one ion makes it more favorable for other ions to essentially join it in the same spatial location, leading to this phase separation, which will eventually get us to this expanded crystalline state. Okay, so this was kind of one of the first things we looked at, but we also wanted to take a step back and really understand the design principles behind these systems. And so the question that we had is, we know that electron transporters, or polymers that are good at electron transport, are very, very crystalline. But polymers that are good at ion transport are very amorphous. And so the question was, how can we get a very crystalline and at the same time very amorphous material? And of course you can't do that, but our design idea was to balance amorphous and crystalline regions to see if we can optimize how fast ions can enter these types of systems. And so we started working with some fairly well-known polymer systems. So this is P3HT, regioregular, which means that the side chains are all kind of on the same carbon for each monomer, and this forms a very crystalline system. Its partner is regiorandom P3HT, which now has our hydrocarbon side chain on different carbons or random carbons on this thiophene backbone, and this leads to an amorphous polymer. And so what we did was we basically took a range of blends of these two different polymers, all the way from 100% amorphous to 100% crystalline, to see how ion injection would change. And so we started by doing a number of cyclic voltammograms, where we can see that the oxidation potential of ions going into the polymer shifts as we basically decrease the crystalline content. Okay, and so the next thing we did was we used a technique called spectroelectrochemistry to basically track how fast ions enter this polymer when we apply a bias. And so essentially what we do at time 0 is we apply a bias, and then we watch how fast ions enter our film. And so you can see if we look at the crystalline film versus the amorphous film, we see a huge difference in the rate at which ions enter the film. So by about 100x in the time constant. But what was really neat is that if we added a little bit of amorphous content to the crystalline film, we actually got faster ion injection kinetics. And so this doesn't look like very much, but this is actually twice as fast as the crystalline film. We can look at other combinations too. They're a bit slower than the crystalline film, but this really showed that by essentially tuning the crystalline character of the material, we can also optimize ion injection kinetics. Okay, and so this gives us our bulk picture. Again, we want to zoom in and see exactly what's happening at the nanoscale in these systems. So again, we're back to our photo-induced force microscope to look at the nanoscale infrared signal. So again, we essentially dope one side of our film. So we have ions on the right, no ions on the left. And then we can basically zoom in and see what the distribution of ions looks like in the doped regions. And so as a control, we just wanted to look at regions with ions and regions without ions. And so if we zoom in to each side here, you can see on the left, this is an undoped region. The topographies here look almost the same, yet if we look in the IR, we don't really see anything but noise. Whereas if we look where there are ions, we see a lot of ion heterogeneity on the right. And so this was really exciting to see. We didn't really expect there to be that much heterogeneity, but it looks like ions are kind of gathering in some kind of clusters together. And how these ions are arranged in the polymer is really going to affect charge transport in this system. And so if we now vary our crystallinity, you can see as we go from 100% amorphous, so 100% regio-random, we don't see too much heterogeneity in our ion signal. But as we start to add these crystalline regions to the polymer, we see these kind of bright spots light up. And right now what we want to do is do some correlative imaging. So kind of basically image the same area with our nanoscale IR technique and with nanoscale stiffness mapping to essentially see if we can correlate crystalline regions to where the ions are going in these materials. Okay. And so for the last few minutes, I just want to transition into talking about a little bit about 2D materials, something we've been excited about for a while now. So looking at this phase transition in these 2D perovskites. So we start off with this crystal structure here, where we have inorganic and organic, basically layered materials. So these work as 2D quantum wells, where we have an exciton that's typically confined in these 2D layers. And as we heat up this material, we see that these alkyl side chains essentially melt, and that changes our lattice spacing and also changes the optoelectronic properties of our material. And so the phase transition temperature depends a lot on the chemical properties of these organic cations in our organic layer. So something like pentylammonium, which has 5 carbons, has a phase transition temperature of about 48 degrees, whereas hexylammonium has a phase transition temperature around 82 degrees. And the question that we wanted to ask here is if by mixing these two organic molecules, if we can tune the phase transition temperature. So the first question is, do these actually mix? And so we can do X-ray diffraction to show that they do. And so we essentially see this is our pure hexylammonium. This is our pure pentylammonium. And you can see when we combine the two, we get essentially an intermediate lattice spacing. If we saw phase separation, what we would expect is this would be 2 individual peaks, but it looks like they mix. And then using DSC, or differential scanning calorimetry, we can actually measure these phase transition temperatures. And you can see really nicely that if we combine pentylammonium and hexylammonium, we have an intermediate phase transition temperature of about 63.3 degrees. And so this was exciting to see. If we blend 2 chemically alike species, it seems like they mix. It seems like we can tune the thermodynamic properties. But what we're currently working on is what happens if we mix 2 cations that aren't alike at all. And so we can do that with this kind of model system that we're working with. So this is phenethylammonium iodide. So it has this benzene ring on it, and this 4-ammonium butyric acid iodide, which we call GABA for short. And so this has this carboxylic acid group here. And so when we mix these, if we look at the X-ray diffraction, you can see that instead of seeing an intermediate peak between these 2 phases, we essentially see 2 different peaks. So this tells us that these 2 cations are actually phase separating when we make these materials. And if we do AFM on these samples, they look something like this. You can see the kind of 2D structure of these materials, but it's really hard to tell what material is what. So which of these flakes corresponds to our GABA and which corresponds to PEA. And so here is again where we can use our photo-induced force microscope to go in and actually see which features corresponds to which chemical species. And if we do that, we get images that look something like this. And now we can essentially say, okay, well, this blue region here is GABA, and this green region here is PEA. And we can do this based on just the infrared modes of these 2 different chemical species. And so what we're really excited to do now is essentially try to tune our properties so we can get kind of different blends of these materials to potentially make things like solution processed heterostructures of these 2D materials. And so in conclusion, I mainly talked about our work on conjugated polymers today. So polymers, we saw can undergo these structural transformations between 2 distinct crystalline phases as we drive ions in and out. We see that these structural transformations impacts ion motion at the nanoscale. We can observe this phase separated state as we start driving ions into our material that looks something like this. And that ion insertion in conjugated polymers is really heterogeneous. And so we think that ions are going to crystalline regions first. We have some other evidence for this, but we're working on some more correlative imaging approaches in order to look at this further. And again, I want to acknowledge my group. So most of this work was done by Seth and Rand. With our funding sources here, we've had a lot of a great partnership with Molecular Vista, and we're always looking for graduate students, postdocs, and summer REU students. Utah is a really beautiful place. This is me at one of our national parks, and I'm happy to take any questions.
Awesome, thank you so much, Professor Bischack. Yeah, that's a lovely national park photo that you got there as well at the end. I would be excited to maybe spend some time there in Utah. But yeah, if anyone has any questions, once again, we'll have a few minutes here to get any questions answered, either in the Q&A or in the chat. So I'll just give that a few minutes.
Hi, Connor. Yeah, a great talk. So I was fascinated by your video of the ion wave moving through the sample. I couldn't read exactly the time scale on the top, but how long did it took you approximately per image?
So this is a pretty slow ion migration. I think each image was maybe 2 minutes or so.
Okay.
So yeah, and here we're essentially parked at 1 wavelength, so we can image quite a bit faster.
Okay, got it. Thank you.
Thank you once again, Professor Bischack, for your presentation today and also for your time.